So these are the reference books, and we'll be starting with plasma membrane. So there are three parts to this topic. First, we'll deal with the structure of plasma membrane, and then we'll deal with the function of plasma membrane, and then we'll study the junctional complexes present. Sorry. Now, if we think of biological membranes, there are some fundamental common properties that all the biological membranes share. All membranes are impermeable to polar charged solutes, but permeable to non-polar compounds. So polar or charged solute means all those molecules that are soluble in water. Most of the biological membranes are 5 to 8 nanometer in thickness. So that is 50 to 80 angstrom thick. We consider the thickness of this biological membrane by taking into account the proteins protruding on both sides. And they appear trilaminar when viewed in cross-section with electron microscope. Now I'll come to the concept of trilaminar when I'll be showing you some membrane pictures. If we look into the development of the concept of the plasma membrane, it started long back in 1926, as you can see. This is the Gauder and Grendel model from 1926. It is the most simplified of model. You can see that these are the lipid molecules of the hydrocarbons, lipid molecules, and the lipid molecules have their polar group facing outside and their non-polar tails facing the inner side of the membrane. This was the simplest of all the membrane structures that came up. So this was in 1926. In 1943, the membrane structure was further developed, and you can again see that the membrane is no doubt made of a lipid bilayer, as you can see. Why do we call it a bilayer? Because there are two halves of the lipid layer. See, this is one lipid layer, this is the other lipid layer. When we imagine a cell, so this is one layer and this is the other layer. So this is the bilayer. And when we imagine a cell, you have to imagine that one of the layer is facing the exterior part, and the other half of the layer is facing the cytoplasmic half. Now, when we move on to 1943, that is the Dawson and the Danieli model. And at that time, it reflected the belief that the proteins present in the membrane were smaller in size. And you can see, so we have the lipid bilayer. The proteins are present on the surface. So they are, at that time in 1943, when Dawson and Danieli proposed this model, they had this concept of peripheral proteins, that is proteins present on the surface of the plasma membrane, and they were small proteins. And we have the lipid bilayer, so that remains constant. And in 1972, the most accepted model for plasma membrane is the Singer-Nicholson fluid mosaic model in 1972. If we look into this model, as I said, this is the lipid bilayer. So there is one layer facing the extracellular space, and this is the other layer which is facing the cytoplasm. And the concept that developed with the Singer-Nicholson model was that proteins are of two categories found in the plasma membrane. One of these category of proteins, as you can see, pass through the membrane. So they travel through the membrane, and these category of proteins came to be known as the integral proteins. And this is a transmembrane protein. You can see it is crossing the membrane and is an integral part of the membrane. This is also another protein you can see which is present, which is an integral part of the membrane. And you see that there are another category of protein which is attached to the surface of the phospholipid bilayer. So these proteins are anchored to the surface of the phospholipid bilayer. So these are the peripheral proteins. So he developed the concept that proteins are of two types that are found on the membrane. One category of protein are the integral proteins that are intricately associated with the membrane. Some of them travel through the membrane, while some of the proteins are present on only one half of the bilayer and is anchored to the phospholipid bilayer. So we have a peripheral protein on the extracellular surface. We have another protein on the intracellular surface. So this was and is the most accepted model of plasma membrane, and this model came to be known as the fluid mosaic model. And this picture, what you are seeing here, the D1, this is a model with the peripheral and the integral proteins shown. It is shown that how the proteins, you can see this is the protein, this is the structure for polypeptide. Okay, so this is a receptor protein they have shown here. This is also a receptor protein, but this is a transmembrane protein. It is crossing the membrane. And these are the peripheral proteins. This is the contemporary model with the peripheral and the integral proteins shown. So this was how the structure of plasma membrane developed through time from 1926 to 2001. Now, this is some more comparison of the, if you look into the Singer-Nicholson model. So this is the Singer-Nicholson model. And this is a model that was proposed by Donald Engelmann in 2005. So these two models are similar, but certain additional features, as you can see, is that in the plasma membrane, as we know now today, you can see certain proteins, certain lipid molecules form aggregates. So the concept of lipid wraps have come up. We'll not get into the details of lipid wraps. Let's concentrate on Singer-Nicholson. Singer-Nicholson model is much simplified, and you can see the protein is spread out through the bilayer. But when we move on to the present version of that Donald Engelmann updated model, you see that we are seeing protein aggregates in between the membranes. So that is one of the modern updated version that was observed, because the lipid wraps are discovered, and it was seen that lipid wraps are nothing but certain portions of the membrane have patches where lipid molecules and proteins aggregate to form a large interacting platform. So let us consider lipid wrap that much for now. This is a very simplified, this is a much simplified structure of the Singer-Nicholson model, as you can see. So we have the proteins. The proteins travel through the membrane. So these are the integral proteins. Or the proteins remain attached to one side of the lipid bilayer. So these are the peripheral proteins. And another thing, if you look into this particular picture, you see that carbohydrates are also present in the membrane, but the carbohydrates are attached either to the phospholipids, phospholipids. So they either remain attached to the phospholipids, so they are present in form of glycolipids, or they remain attached to the protein, so they are present in form of glycoproteins. So this is from this what I have said till now, you have gathered this much information that the plasma membrane is a trilaminar membrane. It has two lipid bilayer, one layer facing the exterior, the other layer or the half faces the cytoplasm. There are two categories of proteins present within the plasma membrane. They are interspersed within the plasma membrane. One category of protein are known as the peripheral proteins, and they remain attached to either of the bilayer. They can remain anchored to the inner half of the bilayer, or they can remain anchored to the outer half of the bilayer. And we have this proteins traveling through the bilayer. So these are the integral proteins. Carbohydrates are also present in the plasma membrane, but they remain anchored to either the phospholipids or they remain anchored to the proteins. If they remain anchored to the phospholipids, they are the glycolipids. And if they remain anchored to the proteins, they are the glycoprotein. Now, the important thing to note here is that when I'm talking about the lipid bilayer, this lipid bilayer is formed of phospholipids. So these phospholipids, which have the consists of a non-polar region of the lipid molecules, which face the interior of the core, and their polar heads, these polar heads of the lipid molecules face outward, interacting with the aqueous phase. The individual lipids and the protein units, if you look into this picture, the individual lipid and the protein units in a membrane form a fluid mosaic with a pattern like that of a mosaic ceramic tile and mortar. So you can see the proteins are dispersed throughout the membrane, and when you look from the top, it looks like a mosaic pattern. There is no defined symmetry to the distribution of proteins in the lipid bilayer. Hence, it is known as fluid mosaic distribution of the proteins. And one more important property is that the membrane is not rigid. It's not fixed. Molecules in the membrane, that is the protein in the membrane, and the lipid molecules in the membrane are capable of moving. They have this sort of flexibility. They can move. And hence, it's called fluid because most of the interaction among the components of the membrane are non-covalent, thus leaving individual lipid and protein molecules free to move laterally in the plane of the membrane. So there is this freedom of lateral movement between the molecules. And since there is this freedom of lateral movement, so it gives the fluid nature. So this lateral movement freedom gives the fluid, brings about the fluid nature of the membrane. This is another picture of the model of 1972. And I showed you this picture to give you the glimpse of how mosaic the lipid looks from the top. So we have this carbohydrate attached to proteins. So these proteins are glycoproteins to which carbohydrates are attached. We have cholesterol molecules present in the lipid bilayer. You can see the phospholipid head, the head of the phospholipid head, and the tails. These are the peripheral proteins. You can see the integral proteins. Now, if you look into details of the integral proteins, these proteins also have, if you just think about it, these proteins also have three domains. One part is associated with the hydrophilic region of the lipid bilayer. The interior part is associated with the hydrophobic region of the lipid bilayer. And again, the part facing the cytoplasm is the hydrophilic region, which is associated with the hydrophilic region of the lipid bilayer. Now, as I said, we have this concept of lipid rafts present in the membrane. So these are areas where a lot of molecules come together, interact, and form platforms. So these lipid rafts are mainly enriched in cholesterol and proteins, GPI anchored proteins. We'll talk about the GPI anchored proteins later on. So they have this lipid rafts have cholesterol and GPI anchored proteins in plenty. And you can see the waviness of this fatty acid molecules. So the inner surface with this wavy acyl chain actually denotes the fluid nature of the lipid bilayer. This is the picture from Leninger, and now we move on to the points of the fluid mosaic model. As I said, the phospholipids form the bilayer, in which the non-polar regions of the lipid molecules face the inner core, and their polar head groups face outward, these polar heads interact with the aqueous phase. So this is the outside of the cell. This is the inside of the cell. The membrane is, sorry. Now, coming to further, now proteins are embedded in this lipid bilayer by hydrophobic interactions between the membrane lipids. So as I said, the protein has three domains. So we have a hydrophilic domain of the protein, a hydrophobic domain of the protein, and again a hydrophilic domain of the protein. So the proteins are embedded in this bilayer by hydrophobic interaction between the membrane lipids and the hydrophobic domains of the protein. Some proteins, as you can see, protrude out from one side of the membrane. So you can see some proteins are protruding out from one side of the membrane, or sometimes they can protrude out from both sides of the membrane, but that gives an asymmetry. Remember, asymmetry means the number of proteins present on the outer surface in case of lipid bilayer does not equal to the number of proteins present on the inner leaflet. So there may be more proteins present on the inner leaflet of the lipid bilayer as compared to the outside. So there is no symmetry between the distribution of proteins on the two sides of the bilayer. Hence, there is this asymmetricness of distribution of the membrane proteins. And another thing that I pointed out was that these proteins, they are of two categories. Some proteins remain associated, anchored to the either to the inner surface or the outer surface, while some proteins travel through the membrane and are an integral part of the membrane. We have these peripheral proteins and the integral proteins. The individual lipid and protein units in a membrane, once more, forms a fluid mosaic pattern and is free to change constantly. And we are seeing that carbohydrate molecules are attached to the phospholipids. So they are either present in form of glycolipids or they are present in form of glycoproteins. And finally, the membrane is fluid because most of the interactions just now, as I said, was non-covalent, as a result of which the reactions being non-covalent, the individual lipid and protein molecules are free to move laterally. This was about the fluid mosaic model of Singer-Nicholson. And these were the points which I just now discussed that was proposed by Singer and Nicholson. Later on, further concepts developed to the fact that the concept of lipid raft later on developed, and the dynamicity, the membrane is a very dynamic membrane, was gradually discovered because the membrane plays a huge role not only in giving a protection to the cell, but it also controls the movement of particles from the interior to the outside and from the outside to the interior. So the membrane is a very important component. Now we move on to the most important component of the membrane, that is the typical phospholipid molecule. As you saw in the membrane, the lipid bilayer consists of phospholipids. Now the phospholipids consist of two basic parts. There is a head and there is this tail. The tail is the fatty acid tail, as you can see, and the head comprises of, this is the phospholipid molecule, the head, here it has been, this is phosphatidylcholine, but in the lipid, we find the presence of phosphatidylserine, phosphatidyl ethanolamine, phosphatidyl inositol, we have phosphatidic acid, and we have sphingomyelin. So the major phospholipids found are again ones of phosphatidyl ethanolamine, phosphatidylcholine, sphingomyelin, phosphatidylserine, phosphatidyl inositol, phosphatidic acid. Here the structure of phosphatidylcholine has been shown. You can see this is the head group, and this is the hydrophobic tail which faces the interior core of the lipid bilayer. And this is found on the exterior surface. This can interact with the aqueous interface on the extracellular surface, or it can interact with the aqueous interface of the cytoplasm. So this is how the phospholipid molecule looks. This is a very simplified diagram of the phospholipid molecule. So this is the hydrophilic head and this is the hydrophobic tail. So I was talking about the major phospholipids. I gave you just now the name of the major phospholipids present in the plasma membrane. And as you can see, we have phosphatidylethanolamine, we have phosphatidylserine, phosphatidylcholine, phosphingomyelin, and fatty acid and the sphingosine. Now, if you look into all these phospholipids, you can see that phosphatidylserine is a negatively charged phospholipid. We have this phosphatidylethanolamine. So this is the structure of phosphatidylethanolamine. This is the fatty acid tail. Phosphatidylserine, sphingomyelin, phosphatidylcholine, sphingomyelin, and sphingosine. Now, in this picture, the different head groups are represented by different colors, and the lipid molecules are phosphoglycerides. So these from A to C, these are phosphoglycerides which are derived from glycerol. When we come to D, this is derived from sphingosine. So this has been derived from this. So this is the precursor. So sphingomyelin has been derived from sphingosine and is therefore a sphingolipid. As I said, a point of note is that the phosphatidylserine carries a negative charge. Now, this particular phospholipid is important in the sense that in your syllabus, there's another topic that we will be dealing with, that is apoptosis. Apoptosis means programmed cell death. Just like a cell has a life, a cell is also programmed to die. Now, how do other cells in the vicinity understand that a cell is dying? Normally, phosphatidylserine is present on the inner leaflet. What do we mean by the inner leaflet? Just keep this in mind. When I talk about the outer leaflet and the inner leaflet, if this is the lipid bilayer, the upper half is the outer leaflet. So this half is facing the outside of the cell. So this is the outer leaflet and this half is facing the cytoplasm of the cell. So this is the inner leaflet. As I was talking about the phospholipid, phosphatidylserine is present in the inner leaflet of the cell. It is restricted to the inner leaflet of the cell. But when the process of death starts within a cell, when the cell is about to die, it has to inform others that I am dying. What happens? This phospholipid, phosphatidylserine, moves from inner leaflet to outer leaflet. So this is a classical way of identifying whether a cell is dying or not. So the moment we see that PS, phosphatidylserine, exposure to the external surface, you have to understand that the process of cellular death has started within that cell. So this is one thing that phosphatidylserine is, it's noteworthy about phosphatidylserine. Phosphatidylserine movement of phosphatidylserine from the inner to the outer leaflet indicates that the cell is on the verge of death. So it is during apoptosis or cellular death, it migrates, it moves from inner to outer leaflet. So phosphatidylserine exposure on the outer surface marks a cell for destruction. So that is a marker phospholipid. It is marking a cell for death. So that was about phosphatidylserine. Now we come to the distribution of phospholipids on the membrane. So this picture is the asymmetric distribution of phospholipids between the inner and the outer monolayers of the erythrocyte plasma membrane. So this is the structure of the erythrocyte plasma membrane. And as I was talking about the different phospholipids present in the membrane, remember, we have two leaflets. One is the inner leaflet, which is facing the cytoplasm, and the other is the outer leaflet, which is facing the exterior of the cell. And if you look into the distribution, what is the amount of phospholipid presence on both sides, you can see phosphatidylserine totally restricted to the inner leaflet, and phosphatidylcholine and sphingomyelin are more present on the outer leaflet of the lipid bilayer. So this is, as you can see, when we talk about phospholipids also, the phospholipids are also not symmetrically distributed. There are some phospholipids which are present more to the inner leaflet, and there are some phospholipids which are present more to the outer leaflet. So there's a asymmetry in distribution of the phospholipids also, just like we studied the asymmetry in distribution of the protein molecules in the lipid bilayer. So when we we have done with the asymmetry of the protein in the lipid bilayer, and we also studied the asymmetry of the phospholipids in the lipid bilayer. Now we are going to another structures which I was talking about, that is the carbohydrates are also present in the lipid. But the carbohydrates do not exist in their own way. So the carbohydrates are either present in form of glycolipids, or the carbohydrates are present in form of glycoproteins. So these are some of the glycolipid molecules. You can see these are some of, this is galactose being attached. This is again galactose in this is basically a structure of a ganglioside. So you can see the structure, a ganglioside always contains one or more negatively charged sialic acid moiety. So you can see this is the sialic acid part. In human cells, it is mostly N-acetyl neuraminic acid or NANA. It's N-acetyl neuraminic acid or NANA, which structure is shown here. In bacteria and plants, almost all glycolipids are derived from glycerol, and as are most phospholipids in animal cells. Now, this is the structure of sialic acid. So you can see in human cells, this gangliosides are present. And since these gangliosides are present, the gangliosides contain this presence of sialic acid. So this is also a big important point regarding the recognition of a self cell from a non-self cell during immune reaction. Sialic acid also plays an important role in helping the immune cells to understand which is a self and which is a non-self cell. So these are the variety of glycolipids. So these are the variety of glycolipids that are found on the plasma membrane. And this is the 4 possible modes for binding of the peripheral membrane proteins. Now we are coming to the proteins. If you remember, I said that the proteins can be of 2 categories. One is the integral and one are the peripheral. So when we talk about the peripheral membrane proteins, they can bind with the phospholipid bilayer by hydrogen bonds and ionic interactions. They can form amphipathic alpha helices. They can form hydrophobic loops, and they can remain associated with the integral proteins. So this is how the peripheral membrane proteins remain attached to the plasma membrane. And this is a point where how experimentally can we differentiate between integral and peripheral proteins? Integral proteins, as you can see, are very firmly associated with the lipid bilayer. They cannot be removed so easily. They are removable only by agents that interfere with the hydrophobic interactions. So since they are a part and parcel of the membrane, so if we add detergents, the detergents interfere with the hydrophobic interactions between the lipid bilayer and the protein molecules, and the integral proteins can be separated. But peripheral proteins, as you can see, are associated either with the external half or the internal half. So these proteins associate with the outer leaflet or the inner leaflet via electrostatic interactions and hydrogen bonding. They can be released easily from the membrane by mild treatments that interfere with electrostatic interactions or break hydrogen bonds. So simply, if we change the pH, the change in pH often results in the releasing of the peripheral proteins. And this is the human erythrocyte membrane. And this membrane is the most convenient, was the most convenient membrane to study the plasma membrane structure. And you can see this is the membrane spanning integral membrane protein that has been shown here. This is the structure of the protein, which is known as glycophorin. It contains an amino terminal. Remember, protein is nothing but a polypeptide. So a polypeptide, so it has an amino terminal which contains carbohydrate chains. You can see carbohydrate molecules on the outer surface. And it has a carboxy terminal which protrudes inward into the cytoplasm. So when I started with saying that the membrane looks trilaminar in nature, what I meant was that, so you can see this is the one layer of lipid. It looks like this is basically a lipid bilayer, but if I consider this to be one layer, this is the layer of protein that is protruding out. This is the layer of protein outside of the cell. And this is the layer of protein that is protruding to the inner side of the cell. So it looks like there are three layers. So that is what was meant by when I said the trilaminar. Looks like a trilaminar. And if we measure this, it is about 5 to, at the very start I said, it measures about 5 to 8 nanometer in diameter, which includes integral protein protruding on the outer surface and the proteins protruding on the cytosolic surface. Before ending today's class, I'll just do one more part of today's class. When we talk about the integral membrane proteins, there are, as you can see, basically 6 types of proteins. If we look into the type 1 and the type 2 protein, these are single transmembrane. You can see the protein is traveling through the membrane. In type 1, the amino group is on the outside and the carboxyl group is on the inside of the cell. So this is the inside of the cell, this is the outside of the cell. In the type 2 category, the amino group is inside of the cell and the carboxyl group of the polypeptide is outside of the cell. When we move on to the type 3 integral membrane protein, you can see this is a multi-pass protein, means this is a long polypeptide, and you can see the polypeptide is traveling through the membrane in this manner as my cursor is moving. So this is a multi-pass protein. In type 4, several transmembrane proteins have become assembled together to form a channel. So this is one polypeptide, this is one polypeptide, this is another polypeptide, this is another polypeptide, this is basically type 5. So type 4 is this one. Sorry, this is the type 4 category. In the type 4 category, you can see the transmembrane domain of several different polypeptides assembled to form a channel. This is type 4. No, I think so the write up here is wrong. This is actually, when I talk about type 5, this is, the picture is not matching with the naming of the picture here is wrong. This is type 4. So this, as I was talking about, transmembrane domain of several proteins assembled to form a channel through the membrane. Remember, the difference between type 3 and type 4 is that in type 3, it is a single polypeptide that is traveling through the membrane many times. But in type 4, different polypeptides come together, pass and pass the membrane individually, pass through the membrane individually. They come together to form a channel. Then we have the type 5 proteins. They are held to the bilayer by covalently linked lipids. So they attach to the bilayer by covalently linked lipids. And finally, we have the type 6 proteins that have both a transmembrane region. As you can see, they are attached, they have a transmembrane region, as well as they are anchored. The inner surface is anchored to the lipid molecule, like in type 5. So it's anchored to a lipid molecule. So these are the variety of integral proteins that is observed within the lipid bilayer. There's another picture. There's a very similar picture. Sorry. So this is the 6 modes of association. So that was how the different integral proteins are present in the membrane. And this is a picture of how the different peripheral proteins are associated with the membrane. For this RAS protein, this is a very famous protein. And when we'll be talking about the topic cancer, we'll again come to this protein, where a mutation of this protein is often seen in patients suffering from bladder cancer. So this protein, as you can see, it's a C-terminal isoprenoid tail. We have a C-terminal isoprenoid tail that attaches the RAS protein to the bilayer. So this is the RAS protein. It is attached to the bilayer. Now this is the SRC. When we will be doing signaling molecular cascades, we'll again study this protein. So this is the SRC peptide. It is attached to the membrane by an N-terminal myristoyl tail. So we have an N-terminal myristoyl tail by which SRC is attached to the bilayer. So there is electrostatic interaction occurring between the acidic lipids and the amino acids. Okay. Next, we move on to C. Where is C? C, we have C. So this is the C-terminal glycophosphatidylainositol tail, GPI anchors, that is anchoring TH1 to the bilayer. I'll be talking about the GPI anchors in my next class. Then we have another one, electrostatic interaction of the phospholipid, which can bind to the annexin. So this is the binding of the annexin protein to the phospholipids by electrostatic interactions. Then E, hydrophobic helices of prostaglandin. So hydrophobic helices of prostaglandin. So this is the prostaglandin molecule. And you can see these are the hydrophobic helices of the prostaglandin, which partially is penetrating into the lipid bilayer. So this hydrophobic helices are partially penetrating into the lipid bilayer. And F, the peripheral protein, this is beta-catenin. Let us look at beta-catenin first, blue and purple. So the peripheral protein beta-catenin associates with the cytoplasmic portion of the transmembrane adhesion protein cadherin. So this is cadherin. This is the transmembrane protein. This is the tails of cadherin. And you see the cytoplasmic protein beta-catenin has attached to the tails of cadherin. So these are the different ways in which peripheral proteins might remain attached to the lipid bilayer. We'll stop our class today.